So yeah, if you want to see if you have a dangerous mutation in your body, the first step is that a doctor would often want to extract the person's DNA. So by extract, it's just taking some cells from the human and taking out the DNA. And once you have that DNA, you can test it in a large variety of ways. So you can test the sequence of the DNA, like read all the letters. You can use some other techniques like gel electrophoresis, and that's seeing some special differences in the DNA. And after testing, like Arpon brought up, you can use gene therapy. So gene therapy is we're gonna cover it later, but it's just trying to fix the problem caused by genetic diseases. So now time for another lab. Oh, sorry, somebody raised their hand. You can go
Yeah,
ahead.
what are we doing right now? Like,
Oh, okay. So well, we just went over the basics first off, and now we're
uh-huh.
gonna do some more stuff with labs. So, like I said just now, um, it usually you can have dangerous mutations in your body, and a
Mm-hmm.
doctor would want to test for those mutations. So now we're gonna go over how doctors can take out DNA from your body and test for that stuff.
Okay.
So if you want to follow along, like if you're using the same device for the Zoom call as you're gonna do for this stuff, then you can click on the links in the Zoom chat and you can follow those. And if you
Mm-hmm.
don't want to follow along, you can just look on my screen and I'll do it too.
Yeah.
So if you want to use this, you need Flash Player. But yeah, so what this is DNA extraction. And it's the first step to seeing if someone has a dangerous mutation in their body. Because you need to get the DNA from a person before you can actually, read it. So in this case, as an example, they're going to be doing a DNA extraction with plants. So really, what it is you just take off a leaf from the plant and then you put it in a test tube. And now what we're going to do is break up the cells. So this step is actually called cell lysis, and lysis means breaking up. And this is the same principle for like human cells. You just take a little sample from the cheek of someone, like take it out from their cheek, and you just, uh, break up the cells. So you can see that they just used a little drill there. And now they're using a special solution. So this is a liquid that's going to break up the cell membrane. So if you've ever looked at a picture of a cell before, you know that it has a coating around it, a protective layer. And that layer is responsible for protecting the cell from its DNA leaving the cell or anything else. But in this case, they want to get the DNA out of the cell so they can start, reading it or, do whatever they want with it. So they added a special solution and they kept it at a hot-ish temperature for a while. And now the goal is to keep the cell parts that they want, which is really just the DNA, and get rid of the rest. So the question is, how do you, once you have all this stuff, you added a bunch of special chemicals to it to break it all up. How do you keep the DNA and get rid of the other stuff? So what they use for that is it's called a centrifuge, and if you've heard of uh, centripetal force, I always have trouble saying that word. Centripetal force. It uses that to separate the heavy stuff from the light stuff. And in this case, the heavy stuff is all the other cell parts, like the cell membrane and everything, and different organelles. And what you want to keep is the DNA, because the DNA is lighter than everything else. So the special machine, it does it for you by spinning stuff around really fast. And with the centripetal force, the heavy stuff sinks to the bottom and the light stuff stays on top. So you can see here that you have a solution with the unwanted stuff, which is heavier on the bottom, and you have a liquid containing the DNA at the top. So now they use a little thing, a pipette, they call it, to take out the solution, the liquid containing DNA. They put it they put it in a different test tube. And boom. Now all they have to do is just get the DNA out of there, and they use this using ethanol, which is alcohol. So they use something called precipitation. We don't really need to go into the details, but what that does is it just gets the DNA out, and you can see something is appearing here. It's like a little strand. It's a string kind of thing, and this separates the DNA. So honestly, this is way cooler if you're actually doing this stuff. But we can't actually have we can't have y'all do this in person.
That's interesting.
It is interesting. Wow, thank you. So this is really just a set of chemical procedures that leaves DNA to in a test tube while you get rid of everything else in the cell. So now at the end, we're left with pure DNA here. So once you get rid of this extra liquid, you have DNA left at the bottom. And now, usually, we did it with a plant in this case, but when you do it with human cells, you have this DNA from a human and you can do whatever you want with it. So, a doctor would normally want to test this DNA to see if it has some dangerous diseases. So, now there's a bunch of ways you can test diseases. And the main way that they use nowadays is DNA sequencing, where you test the sequence letter by letter of the DNA over here using some more chemical techniques that we won't go into. But what they can also do is they can use something called gel electrophoresis. So, so Arpan is asking, is DNA a protein made of amino acids? So, I mean, I guess that's a good question. No, DNA is not a protein. It's something different. So you can think about it like the it's a completely different molecule. It's a different chemical structure. And if you think about it, the activity sheet made of paper, like if a teacher gives you a list of activities, or sorry, a list of instructions for an activity, it's written on paper, it's completely different. And the activity is to make something out of blocks. So the blocks are completely different than the actual activity sheet that she gave you. That one is made out of paper, and the thing that you're making is made out of blocks. So that was really jumbled. I don't know how you understood that, but yeah, it's completely different. It's more like the instructions on how to make proteins than it actually is a protein itself. And yeah, this is it's not a real picture of the DNA itself, but it's a real picture of the experiments, the experiment results that you get from here. So this is a picture of a special pattern that you get when you do something called gel electrophoresis. So well, gel electrophoresis is just a technique to look at how someone is at. Well, it's really to look at the differences between different people and how their DNA is, what their DNA sequences are like. So if you have a special sequence of DNA, then this little pattern is going to look differently in your case than in another case. So this number 5 is one person, and this number 8 is another person, and you can see that it looks different here. So we're going to talk about how you actually get these results. But in this case, this special pattern indicates that someone is at risk for some disease, as you can see here. So, well, it's not actually a disease in this case. It's more like it's a condition that's not that serious. But in many cases, these different patterns of bands are actually they're showing if someone is at more at risk or less at risk for a certain disease based on their sequence. And this is what they do if they don't really have the time and the money to do actual sequencing on someone's genetic data. So again, this is called gel electrophoresis, and you can just click that link in the Zoom chat if you want to follow along. So this is just, we're going to go over the general procedure of how you do that. So look at what we're starting with first off. We're holding a little test tube with some liquid containing DNA inside it. So, well, if you look at what we ended up with DNA extraction in the last thing that we did, it's a test tube that is filled with DNA. That's a very final product of our last thing. In this case, the beginning product is a test tube filled with DNA. So after you do DNA extraction, you can go ahead and do this gel electrophoresis, and you can figure out if someone is at risk for disease. So in this case, it's I don't know actually what it is in this case, but it's just a bunch of DNA strands in there, and they're all of different lengths. So some of them are really short, some of them are really long, and actually the reason that some of them are short and some of them is long is because once you extract the DNA, it's really long. Okay, so it's an insanely long thing. It's a really long molecule, and we can't really use it for anything. You can't use that for sequencing or anything like that. So what doctors do after they extract DNA is they chop it up with a special enzyme, which is just a little chemical called a restriction enzyme, and that chops it up into different sizes. So it chops it up into short, medium, and long fragments that are all of different lengths. And that's why in this case we start off with a bunch of short, medium, and long fragments. So if you want to sort the DNA strands from shortest to longest, it can really tell you a lot about the different the differences between people's DNA. Because when these enzymes cut, like, let me just show you. It's not in this PowerPoint. I don't know why it's not. But restriction enzymes, they cut in really specific places. Okay? So the way they chop up DNA is really important. So, 1 second. Okay, here. So this isn't really specifically pertaining to this case, but if you have a special enzyme that chops at this little portion right here, or this little portion, then you can see that these are letters just like in DNA. So if you have restriction enzymes, they're just like scissors and they chop at very particular places because that's what they do chemically. And if they, based on how they chop someone's DNA up, then it's it tells you a lot of information about the DNA sequence. And the way they chop it up can be measured by this little procedure that we're going to do. So this is why we need to sort DNA strands by length. And this is what they use gel electrophoresis for. So yeah, they use gel electrophoresis whenever they need to sort DNA strands according to length. All right. So usually, well, the first part of this, let me go over the name first. So the first part of this name is gel, and that's because all of this is happening in a gel. And the gel is special because it's like it has a lot of tiny holes in it, and we're gonna see why that's important in a second. So electrophoresis essentially means separating. I think phoresis means separating, and electro is using it with electricity. So first, let's look at this. So this gel has a lot of tiny holes in it. And while this little animation is happening, um, the gel, you can think of it like a dense forest. So it's a forest with a lot of trees in it. So the principle of this is that you have DNA, which is like people running through a really dense forest of trees. So the gel is like the trees, and the DNA are like the people. So if you have a really big person trying to get through a forest just by running through it, then it's going to take really long for them to get through the forest because they're going to be blocked by all the trees. But if you have a really tiny, like skinny person or a short person, then they can get through it easier because they're not blocked by the trees. Like they can get through quicker. So that's the principle of this. The DNA, we're trying to use electricity to get the DNA to be attracted to this end over here. And there's smaller DNA, which means the shorter DNA is going to go farther, and the longer DNA is not going to go as far because it's going to be held back by this gel, which has a bunch of holes in it. And um, yeah, you can see here that you have an electrical field. So electrophoresis, the electron in this comes from an electrical current. So what they do is they have an electrical field set up, and this side is positive, so it attracts the DNA, and DNA is negative. So DNA goes away from this end and it goes towards this end because negative is attracted to positive, like opposites attract. So you can see right here, DNA is moving through the gel. Yeah, so the really long DNA doesn't go as far, but the shorter DNA can move farther. So again, if you just saw the animation in the top left with the DNA moving through the holes in the gel, so if it's a really long DNA strand, it's going to get held back and it can't move as far. But if it's short, it's going to move faster. So now, this ending part isn't as important, but oh, sorry here. Yeah. So this ending part isn't as important. They're going to show how you do everything from scratch. But what you can see here is that you end up with a really cool pattern. So in each of these little wells, that's what they call them, in each of these little holes, you put a little sample of DNA, you put the electrical field, which makes it go towards this end over here, and that makes the DNA strands all separate. So some of them go really fast because they're shorter, and some of them stay back because they're longer. And what that tells us is just how the restriction enzymes, the scissors, chopped up the DNA in the very beginning. So I'm sorry I didn't show you that step, but really that's important to showing the genetic sequence, because a certain sequence is going to tell the scissors, which is the restriction enzymes, to chop in a certain way, but another sequence is going to make them chop in a different way. So that's why if you have different people and do this to their DNA, like chop it up and run it through this gel electrophoresis, the patterns are going to look different, because some people have uh, chopping patterns such that some of them are longer and they have more clusters over here. And some of them are going to be chopped in a way that they have more shorter fragments, which makes them go over here. And again, that's just because the restriction enzymes, they chop at very particular places. So if you go back over here, this is a real picture of like DNA being run through this procedure, and this is what they see. And again, this is a real life scale. So this is actually what the size of the experiments are. This isn't like microscopic or anything. I just wanted to clear that up. So if a doctor looks at this, they can see that these three people have a similar genetic sequence. And if they know that person 3 has some genetic disease, then it's more likely that person 4 and 5 also have that disease. Because after you run it through this procedure, you see that they have a very similar pattern of what things end up long and short when they're chopped up by the enzymes, which means they have a similar sequence of letters in their DNA. So this is much shorter than genetic sequencing, which is running DNA through like a really long procedure. So they usually use this if they want to save time. So any questions before we move on? I know I covered some stuff and I may not have been too clear about it. But if you have any questions, you can unmute now or type it in the chat or anything. All right. So yeah, feel free to ask a question at any point. So now we're going to talk about some other basic techniques of biotechnology. So now we're going to get to some of the really cool stuff like cloning, stem cells, and CRISPR-Cas9. So I don't know if any of you have heard about this stuff before, but if you have, then it's cool because you get to learn more about it, hopefully. So the very first thing we're going to talk about is gene cloning. So you may have heard the word before, cloning, as in making a copy of like a human or making a copy of a sheep or anything like that. But in this case, this is much more widely used, and this is really important, even if you may not have heard of it before. So what gene cloning is, so fundamentally, cloning is making a bunch of copies of something. So cloning is just, a gene cloning is going to be making a bunch of copies of a gene, as you may have guessed. So this gene can be something really important because it can be a gene that codes for a very important drug. And one case that I know is that people who don't have enough growth hormone is just a chemical that makes people grow bigger and taller. People who don't have enough growth hormone can get injections of a special drug that helps them grow more. And this drug is produced, it's not really a drug, it's more of just a growth hormone produced by a gene. So gene cloning can help people produce drugs that are important for them to, survive or something like that. So what this is usually a gene. We call it a gene of interest because it's a gene that we want to copy multiple times. Um, they insert that gene into other cells. So these cells aren't human cells or anything. They're just random cells. And what cells do is they make copies of themselves, and they make whatever product their DNA tells them to do. So this is like, let's say a kid, let's say what he really wants is a Lego toy. Okay? He really wants a toy, a Lego toy, but he doesn't know how to get it. So he wants other people to make it for him. So he goes into the teacher's classroom and he takes the activity instructions that the teachers gave to all the students, and he switches it out for the instructions to make a Lego toy. So now what the kids are going to do, the kids are all going to make Lego toys because all they're doing is just following the instructions that the teacher gives them. So now the kid who switched out the instructions gets what he wants. So in this case, the instructions are the DNA. And what we're doing is we're inserting this gene into other cells, random cells. And they're putting this gene into the DNA. And now what these cells are going to do is we're just going to grow them for a while, and these cells are going to produce the protein because of corresponding to this gene. So the gene, again, is just like an instructions sheet, and the protein is like the Lego toy that's made from following those instructions. So the cells have all made, because it's in their DNA now, they insert this into the DNA, the cells have all made whatever protein we wanted them to make. And then in the same way as before, as we showed you before, using some special chemical techniques, you can isolate some stuff with the centrifuge. They're going to do the same thing to take out this protein from the cells, and they're going to do whatever they want with it. So usually this protein is a special drug that's made by these cells. That's the instructions that they're following to make a drug. And this drug can be, it can be just extracted, and it can be injected into other people to help solve their problems. So here's a little quick lesson thing on gene cloning. So if you guys want to know how to do that, well, the general principles of it, you can click that link. You can watch me do it. So here you can see. Oh, so Arpan was asking what are stem cells? All right, we'll get to that. That's the next thing that we're covering, the very next thing. So here, um, this is just like a general presentation kind of thing. What gene cloning is, really, it's manipulating the organism's genes. So by organism, I want you to think of like the students who are following the instructions, like from the teacher. So the students are like the organisms, like you're just using them to make what you want. So these organisms can usually be a bacteria that are producing some important thing like insulin, which diabetics have to take, or plants, really anything like that. So to clone a gene, what you need to do is put that gene into some other organism, which can make it for you. So you're gonna grow them on a plate just like this. And you can see the sorry, you can see this little part in red, and that's gonna be like a little gene. Like I said before, a gene is just a little segment of DNA. So this entire blue thing is some organism's DNA, and this red part is a gene. So what genetic engineers are gonna do is they're gonna take this gene and they're gonna put it into other organisms. So this is what bacteria DNA looks like. It's not like our DNA where it's just jumbled up. It's a really neat little circle. And usually DNA in bacteria is put into these circles. So they have a big circle that they store all the DNA in, and they also have little circles called plasmids. So these are way smaller than actual DNA, but they're like little circles of DNA that are stored in bacteria. And these circles can contain important things like usually they only contain a few genes, like 3 or 4 genes. But yeah, here, so these little arrows each represent like 1 gene. So it's not really important what the specifics of these are, but like we said, this is called a plasmid. And what we're going to do is we're going to take this bacteria plasmid and we're going to put whatever gene we want into it. So that's a little advanced. Don't worry about that stuff. All right, so if you remember earlier, a few minutes ago, we talked about restriction enzymes. So a restriction site is really just a sequence of letters that's recognized by one restriction enzyme. So we talked about restriction enzymes earlier because we're talking about it chopping DNA up into long and short fragments based on its sequence. What we're going to do here is we're going to use the same restriction enzyme, but we're going to use it for a different purpose. We're going to use it so we can chop this plasmid in the middle, this little circle thing, and we can put whatever gene we want into it. So it's like you use scissors for a bunch of things, right? You cut bags, you cut paper. It's the same thing. Restriction enzymes are just chemicals used by researchers to do a bunch of different things. So you can see here that each restriction enzyme has a little set of letters that it recognizes. So GGATCC. Well, actually, it's GATC, sorry. But that's a specific sequence recognized by one restriction enzyme, which you're going to use. So it's going to be like a pair of scissors that just cuts it wherever it sees that little thing. So it's like in a class, you cut wherever you see a dotted line. And in this case, this enzyme is going to cut wherever it sees this sequence of letters. So boom, it just cuts there and it breaks this little circle apart. So now what it looks like is this. On a larger scale, it's broken open. And I just want to remind everyone that this is a little circle called a plasmid, circle of DNA. And we're manipulating it now, we're cutting it open and everything so we can insert this into bacteria later. We're going to talk about how to do that. So usually what they do, they don't do it one by one. They have everything in a test tube, like you can see here. They have all the plasmids in a test tube so they can put it into bacteria later. So these are really tiny. They're showing it as big circles here, but they're really, really tiny. You can't see it with your naked eye. So now we talked about the plasmids, which is what we're going to use to deliver this stuff into the organisms like bacteria. But what we want to do first is we want to get the gene. First, before we insert into bacteria, what we want to do before that is we want to get the gene that we want to insert into that plasmid. So this is using the DNA for, we're not really sure what DNA it is specifically, but this is the DNA for whatever thing we want to insert into that. We want to take the specific part of it that's coding for the gene we want to do. So if we want to make a drug like insulin, this is the specific part of the DNA that codes for insulin. So we use the same restriction enzyme to chop it up. So we chop it up into tiny pieces, and it's chopped up in a very particular way. And because we use the same restriction enzyme, we end up with these things called sticky ends. So it's the specifics aren't really that necessary, but it's cut in such a way that this binds to this. Because we cut it with the same pair of scissors, this is just inserted into the middle of this. So it's bound together. It's like you're cutting something out and gluing it onto here. That's really what it is. And if you heard the word recombinant DNA before, this is what it is. Recombinant DNA is when you combine, you may see that word in here, when you combine some different segment of DNA with a plasmid, a little circular part of DNA. So we did this on a test tube. This doesn't have anything to do with like actual organisms so far. What we're going to do now is we're going to insert these into bacteria. And we're using bacteria because bacteria are really good at producing these things. And it's easy to get the things into them in the form of plasmids. So this is a solution containing a bunch of bacteria in this little liquid, so you do not want to drink that. So this is just a bunch of bacteria in here along with a bunch of little plasmids. So they're the little circular things. So what we want to do now is get the bacteria to take this into their nucleus. Oh, sorry, not nucleus. Sorry, dude, I haven't slept enough. But we want to get it to take it into their cells. Bacteria do not have nuclei. But what we're going to do now is use a special machine that uses electricity to get the bacteria to take that plasmid into their cells. So now they can produce whatever thing we want because it's inside their cells now. So you put it into this little thing, we put it in this machine, and we're doing something called electroporation. So that uses electricity to create pores. So pores meaning little holes in the bacteria's walls. And it created holes so that the plasmids can pass through it. So the little circles we were working with earlier, they're just taken up into bacteria, and bacteria can read these, and we do whatever we want with them. So now we take this out, which is a solution of bacteria with these plasmids inside them. And then we spread them around on a little plate. So now they just grow and they produce whatever we want them to produce because they're following the instructions on this DNA here. This part is not significant. So we use a special thing with an antibiotic to keep only the bacteria that have this thing inside them. But what we do now is we just let the bacteria grow. And because they're following the instructions in this DNA right here, which we've inserted into them, they're going to produce whatever gene that we inserted into this little thing here. So the gene could code for a drug, a protein that's really important for people to survive, like insulin. It could code for insulin that's important for diabetics. And as they grow, they just produce insulin. So once these things grow, we can really extract the insulin from them or the drug from them. And that's all we have to do. So what we've done is we've taken this gene, inserted it into an organism like bacteria, and we've cloned it. As the bacteria grow, they make multiple copies of that gene and they create proteins. And the proteins are what we want in this case. They're like the final product. So now we're going to talk about stem cells. So stem cells are, well, essentially when you're an embryo, like before you're born, when you're really young, you have cells that can make a bunch of different things. Because when you're in the embryo in your mother's womb, you don't already have like, and for most of that part, for most of that duration that you're in the embryo, you don't already have fully developed like a liver or red blood cells or brain cells or anything like that. You start off with these things called stem cells, and a stem because it's the stem of a bunch of, like a stem of a plant, for example, like a stem of a tree. It starts at one thing and then it branches off into a bunch of other things. So they're called stem cells because they start at one type of cell, which is called a stem cell, in the embryo, which is why it's called embryonic. It's before you're born. And it branches off to produce a bunch of different types of cells. So the reason that this is important is that we start off with these stem cells and we can use special signals to make them produce whatever other kind of cell we want. So let's say someone really needs a liver transplant. Like their liver is failing, it's gonna fail soon. And we need to grow a new liver for them. So what they're doing now with stem cells, what they're trying to do is they're using techniques that can actually help us use these stem cells and produce new liver cells. So it lets us grow a liver just in a petri dish or anything like that. It grows a liver from scratch using these special stem cells. Yeah, Arpan is asking, well, I don't actually understand what you're asking. He's saying, can stem cells be used to cure diseases of other cells? Well, what do you mean other cells? Wait, Arpan, what do you mean like other cells? Okay, I'll let you type it in the chat. So yeah, so you can use it for like liver failure. If you use the stem cells to produce liver cells, you can use it for um, bone cancer. If you need new bone marrow, if you need new bone marrow cells or bone cells or something like that, it's a really exciting promise. So you can use these cells that are usually produced by embryos, and you can create all kinds of different cells. The problem is we can't just take cells from embryos that haven't been born yet, right? We have to take cells from like actual adults because we can't just take cells from babies because that's like unethical. So what you actually have to do is you have to use some special things um, to make regular human cells back into stem cells. But that's a little bit of a nuance. We're not really going to go into that. So this little lesson here tells you about stem cells and all right, Aditi has to go. Bye, Aditi. So yeah, Arpan
Bye.
is asking, okay, yeah. So if someone is blind, for example, yeah. So stem cells can definitely be used for that. So if someone is blind, like because their eye cells are misfunctioning or something, um, theoretically, stem cells can be used to create new eye cells that work better. So stem cells can really like produce any cell in the body. That's what's special about them. So if you can find a way to use these stem cells and produce new organs that work better than other ones, then that's a really exciting thing because you can treat diseases that happen if your organs or your cells malfunction. So yeah, so stem cells are really just basic cells that can produce any other type of cell. So we're not gonna go too deep into this. But yeah, most stem cells are coming from embryos, which are like before you're born. It's like what produces you. It's made up of only a few cells. And these are the stem cells. So these can produce any cell, any type of cell. Because when you're in this stage, these are each individual cells. You don't have a liver yet. You don't have bones yet. These stem cells are responsible for producing like literally anything in your body. And that's why they have to be so diverse, like produce like a diverse range of things. So for example, here you can have like a brain disease. So I think So what we're gonna do now is going to a bunch of examples, just a few examples of how stem cells can be used. So ALS, you may have heard that before. That's the disease that affected Stephen Hawking when he was alive. It's what made him stay in a wheelchair for the last part of his life. So the problem with ALS is that the nerves don't connect correctly between your spinal cord and your muscles. So you can't control your muscles as well. And because you can't control your muscles as well, it results in something called atrophy. Just makes your muscles weaken over time. So you can't function your muscles as well. Your muscles can't function as well. Sorry, I don't have enough sleep. But yeah, the main cause of death is that the diaphragm, the muscle that helps you breathe, doesn't work as well. So they can't breathe. So the hope is that you have these neurons, which are a problem, and you can hopefully find a way to get new neurons from stem cells that work better. And this helps you control muscles with your brain, and that prevents the damage that occurs with muscles weakening. And this is something Arpan was talking about. You can have your eyes getting damaged, and macular degeneration is a disease where a specific part of your eye called the macula is just being destroyed. So you can replace these cells in the macula with new cells produced from stem cells. So you inject those into the back of the retina, or here, and it replaces these cells that are deteriorating. And you just grow these new cells in like a petri dish or something like that. So that's why stem cells can be so special. And now the last thing is CRISPR-Cas9. So CRISPR-Cas9 is a technology that's really growing in current times. So like we said earlier, if there's a way to like correct little errors in the DNA, like here, it's a sickle cell anemia, which you talked about earlier. Like if one letter is wrong in the DNA, you would really want a way to correct it, right? So there's a lot of diseases that happen because one letter or a few letters are wrong in DNA. And CRISPR-Cas9 is a new technology that allows people to hopefully like research new ways of changing the DNA. So like a genetic disease with a few letters wrong can be changed so the DNA is correct again. So this is probably the last lesson that we're doing in this call, and I have another link for you guys after that. So you can just click on that link in the Zoom chat if you want to follow along. So I don't know how long this will take to. Yeah, this takes a long time to load. So yeah, essentially what CRISPR-Cas9 is it's using a special molecular machine. It's the size of this. It's really, really small. And what it can do is it replaces bad DNA with good DNA. Or it can also delete little parts of the DNA. It can do a whole bunch of things. So and that ties into something important called gene therapy, which we're doing in these, which we're trying to develop in these days. Gene therapy is developing developing genes that are better than the genes that you have in your DNA. And by better, I mean like it doesn't have a disease, or it's healthier. Like if you're going blind, you can have a gene that's wrong, and that's the reason you're going blind. So gene therapy is where you try to create a new version of that gene that lets you see, and you deliver that into a cell through like injecting it into like the back of the retina or something like that, and it creates it lets your cells use the correct version of the gene instead of the wrong version, something like that. So let's see if that CRISPR thing has loaded yet. There we go. So let me share my computer audio.
Science is
okay. You guys hear that?
Science is a serendipitous endeavor. We're exploring nature and trying our best to get at the truth.
Chat, okay?
And every now and then,
Yes.
we're surprised.
All right,
When I first heard it, I thought it was too good to be true because, there have been many iterations through my career as a geneticist where people said, oh, we have this method that allows you to change DNA anywhere you want. I thought, well, I've heard that before.
In the simplest terms, CRISPR is a bacteria's version of your immune system.
In the laboratory, we've adapted that bacterial system in a way that we can use it to reprogram genes within our own cells.
I like to use the analogy that CRISPR-Cas9 is like a word processor for our genome. So it's almost like a cursor where you can move it from letter to letter in your DNA.
Until very recently, it's been very difficult to do operations like cut, copy, and paste.
The promise of CRISPR is that you can change DNA in any way you want.
We can literally ask, how do all genes in the genome, all 20,000 genes, how do they influence this one interesting disease or this one interesting facet of biology?
By using CRISPR, we can develop products and novel crops that won't have been possible otherwise.
Discovering drugs, detection, or therapeutics.
The ability to change one base pair at a targeted site in a living cell is something that I think even 5 years ago would have sounded more like science fiction to me.
It's hard to imagine all of the ways that it will be used going forward.
In my 20-year career as a scientist, this is the first time something so transformative has happened.
CRISPR is insanely cool. Very, very big breakthrough.
All right, yeah. So I don't know how much of that you guys got. Somebody said it's glitching, so that's unfortunate. But yeah, what CRISPR is, like they said in the video, is it's taken from bacteria, and it's like a machine that's taken from bacteria, and bacteria use this as part of their immune system. But what we use it for, this new purpose, is to change the way that genes, to change the sequence of genes. So it's like Microsoft Word, if you ever used that before. You can click it, you can edit it, you can do whatever you want. So what CRISPR is it's a little machine like this. And it has some little different parts to it, which we're not going to go into. But it has a little part of this guide RNA, and that helps it detect the correct sequence in the DNA. So, yeah, someone raise their hand. Go ahead.
It's 6.
Oh yeah, it's 6 o'clock. So it looks like we got to go now. I'll just finish this little thing up, and yeah. So Saloni sent a Google form, so you can listen to this and you can just finish this up before you go. But click on that when you're about to go and fill out that so you can get your points for the leaderboard. But yeah, the way CRISPR works is it just binds onto the DNA, it chops it up like a little it unzips a zipper like DNA. And it chops it up like scissors pretty much. And it can break the DNA, it can insert new stuff into the DNA. Like you can see here, it's a mutation, it's a new mutation that I inserted. And this is essentially like a repaired sequence. So if something is wrong, then you can use this little machine to change the DNA sequence. So I think, yeah, that's all for CRISPR. I think that's probably all for today, actually. So I have one more thing that I just want to show you guys in case you want to do it at home. So if you want to, you can do this really cool thing on gene therapy at home. And just look over it. It's like a video. And it shows how gene therapy is applied in the real world. So that's all these techniques being used in the real world to help somebody's disease be cured. So Arpan is asking a really good question. So can using RNA help simplify gene therapy development? So yeah, they actually use RNA a lot in gene therapy. They use RNA to, it's called RNA interference. It's a little bit complicated, but they use RNA because RNA is just like DNA. They use RNA to destroy existing DNA or RNA in a cell. So I don't know about gene therapy development, but it helps to get rid of old genes. Because when you want a cell to express a new gene that helps them see better or something like that, you want to get rid of the old version of the gene. Yeah, bye, Arjun. So RNA would be easier than DNA, right? Yeah. So the problem with RNA is they actually do use that. They use RNA they insert RNA into a cell so it can read it directly into proteins because the pattern is DNA to RNA to protein. But RNA is destroyed really quickly, so it's usually destroyed in the cell. Except if you insert DNA into the cell, it stays there permanently. So if you use RNA, you have to insert RNA again and again, like every week or every day or something like that. I don't know. Yeah, so it's hard for RNA to get to the right cell. And it's really important for DNA to get to the right cell as well. That's just the problem that they have to solve. they use some special mechanisms to get the RNA into the right cell using some cell membrane markers. But yeah, we won't really get into that. So yeah, it is 601. So if you want to, you can click on the link I sent with the gene therapy thing, and you can do that at home to see how it's being applied in the real world. But yeah, you guys can go now. Just be sure to click the link on the forum and fill out the attendance thing. And also, Saloni sent some new challenges that we have on our website, so you can do those and get a lot of leaderboard points. And yeah, she sent the links to those. I'll just send it again, copy paste it here. So be sure to click on those and check it out. So yeah, you guys can go now. Thanks for sticking with us. All right.